Hello friends, welcome to the world of chemistry. This is Dr. Vikram Panchal and I'm dealing with spectroscopy, and now we'll start UV spectra. This is lecture number 8. So first of all, let us understand how UV spectrophotometer, which is the instrument used to get the UV spectra, works. Okay, so let us see the schematic diagram of UV spectrophotometer. So first of all, we take two different lamps. Okay, one is a source of visible light, another is a source of UV light. So these are lamps which produce light. Okay, and this light will be in all directions because they travel in all planes. Then these lights should be focused and pass through a slit or a filter. Okay, so this is a slit, and when this light comes out of the slit, it will be a beam. This is a beam. So main function of this slit is to produce a beam of light. So this will produce a beam of light, a focused light which is coming out of this. Now this will pass through a filter, and main function of filter is to release a light which is having only one wavelength. This will release a light of different colors. So it can be prism also, because prism will split light into different colors. So colored light will come out through this. Now we have to keep one very important filter which is known as slit also, and it is basically monochromator. The main function of monochromator is to allow a light having only one wavelength. So only one wavelength, particular wavelength light comes out of this, and this light is called monochromatic light. This light is known as monochromatic light. That means a light of only one color or one wavelength. So this will produce a light which is having only one wavelength. So that is monochromatic light which comes out of this. Now here there is very important part of this spectrophotometer which is known as beam splitter, and main function of beam splitter is to split this beam in different directions. They will come out. Again, there will be compartments of mirrors. So these are basically mirrors. And main function of mirrors is to split beam. So this is also known as beam splitter. Okay, so this beam splitter will split the beam of the same light having the same intensity, and these two will pass through two different compartments. One is known as reference, another is called sample cell. Main function of this is to pass this light through solution. Okay, So reference cell mainly contains solvent in which the solution is prepared, and this mainly contains solution, and the light will pass through this. The light which passes through this having certain intensity, and we call it I0. That is incident light. Intensity of incident light is I0. So I0 means intensity of incident light. And then this light is allowed to pass through both these compartments, and then they will pass through detectors. Okay, detectors, then they will go through data processing instrument, and then we will get a graph. So here the light which comes out from solvent as well as solution is called transmitted light, and intensity of transmitted light may be I. So this is transmitted light. And intensity of transmitted light is I. When we plot percentage transmittance versus concentration of solution, then first of all we will have to take solvent in sample cell. That means light will pass through reference cell as well as sample cell, but sample cell having solvent only, so there will be no absorption of light, and the same light will come out. Okay, and therefore percentage transmittance could be written as like this, that percentage transmittance is equal to I upon I0 multiplied by 100. If I is equal to I0, then I0 cancel and it will be here. So the first line would appear over here. The first point here, it will be here, where there will be zero concentration. Matlab, there is no solute particles in solvent, and therefore we will get zero concentration over here, and the first point will be here. Now in this sample, let us keep 25% solution. Okay, now 25% solution, so obviously this light which passes through this solution, which is having 25% solution, will be absorbing certain amount of light. Okay, and that may be I absorption. That means I intensity of transmitted light will be less than intensity of incident light for this solution, and therefore there will be certain amount of light which is being absorbed, and therefore transmittance will be less than 100%. So here I is not equal to I0, and if I is less than I0, this value will be less than 100%. So it might be here somewhere for 0.25% concentration. That means 25% concentration having this much transmittance, percentage transmittance. Now we will take 50% transmittance for solution. Obviously for that absorption of light will be more, and therefore I will be even less than I0, okay, or even less than this I, and therefore this may be somewhere here. Okay, then we take 75% solution. As concentration of solute particle increase, absorption of light will increase, therefore intensity of transmitted light will decrease, and so that we get percentage transmittance even less. So this way, if we plot a graph of all this point, then it will appear like this, and this is called exponential increase. Okay, or exponential. Change will be exponential, increase or decrease, which is in fact like this. So this kind of graph appear because of different concentrations of the solution. Now we want a different kind of graph, not this one. And for that, we need certain laws which govern the UV visible spectrophotometer. And therefore, we will start Lambert's law. Okay, so Lambert's law is basically for sample of different thickness. Okay, so Lambert's law is basically for sample of different thickness. Another law is for different concentration, and that is called Beer's law. Okay, so I will take this Lambert's law and Beer's law in detail in next lecture. Thank you so much.